Welcome to Advanced Human Anatomy and Physiology. This is Chapter 2, which is the chemical basis of life. This is a basic review of what you probably have learned or should have retained from biology and also some of your chemistry skills if you have already taken chemistry. If you are taking chemistry concurrently with this course, then you will visit some of these topics early on in your chemistry course. So we are going to focus on three main sections in Chapter 2 as we study the chemistry of life. We'll do a basic introduction in Section 2.1. In Section 2.2, we're going to learn about the structure of matter. And then in Section 2.3, we're going to look at the chemical constituents of cells. And the chemical constituents of cells would be those biologically important molecules that come together to create the fundamental unit of life, which is the cell itself. So in Section 2.1, our basic introduction here, what you need to know as a learning goal is to be able to give examples of how the study of living materials requires an understanding of chemistry. And in doing that learning outcome, we ask this basic question: why on earth would we want to study chemistry in an anatomy and physiology class? So take a moment and think about that. At this time, you might want to pause the lecture just to kind of look back and understand what you've learned in chemistry and perhaps maybe what you've learned in biology and figure out how would chemistry play a role in anatomy and physiology. Again, I say at any time during the lecture, as you go to copy down the notes, you can pause the lecture as the slide is switched. Pause the lecture, and then you could jot down the material on the slides, and then you can play the lecture to listen as what is being said. And if during that lecture component you hear something that might be of value to you for your notes, you could jot that down on your note page where it would fit in. So, why study chemistry in anatomy and physiology class? Let's look at the answer here. Well, in order to understand anatomy and physiology, you need to understand chemistry, because a lot of what we do in biology, especially at the cellular level, has that chemical foundation. So body functions do depend on cellular functions, and this ultimately would come to metabolism. And when we talk about metabolism, we're looking at the chemical reactions of an organism. So cellular functions result from chemical changes in a cell, and biochemistry, which is the chemistry of life, is going to help us to explain those physiological processes. So in order to understand chemistry, we first must understand what matter is and what the structure of matter is. So in Section 2.2, we have a few learning outcomes. The first would be to be able to describe the relationship among matter, atoms, and compounds. Then we should be able to describe how atomic structure determines how atoms interact. Our third goal would be to explain how molecular and structural formulas symbolize the composition of compounds. Fourth, we should be able to describe the three types of chemical reactions. Fifth, we should be able to describe the difference among acids, bases, and buffers. And lastly, in this section, we should be able to explain the pH scale. So those are our six. Those are our sixth, and actually I missed one there. No, I got them all. I just counted incorrectly. Those are our 6 learning outcomes for Section 2.2. So, what is matter? Well, in order to understand what matter is, first let's define what chemistry is. And ultimately, chemistry is the study of matter. Biochemistry is the chemistry of living organisms. So we know that matter is anything that has mass and occupies space. That could be solids, liquids, and gases. So when we talk about mass, understand that mass is the quantity of matter in an object, and volume is the amount of space occupied by that object. Mass is different from weight. So mass is the quantity of matter in an object. Weight is the force of gravity acting upon an object. And we know that matter, it is composed of elements. So elements are composed of chemically identical atoms. We have those elements that are bulk elements required by the body in large amounts. Then we have trace elements in the body which are required by the body in small amounts. And then there are those elements that are ultra trace elements, and those are required by the body in very minute amounts. Ultimately, elements are those things that are composed chemically of atoms. And an atom is the smallest particle of an element. So let's look here. So we have an atom, and the smallest particle of an element that has the properties of that element would be that element's atom. And inside the atom, there are two basic types of subatomic particles that have an actual charge. We have the electron and the proton. The electron is the extremely small particle with almost no weight. It carries a negative electrical charge and is in constant motion around the nucleus of that atom. Then you have the proton, represented with a P+. That's a relatively large atomic particle, and it carries a positive electrical charge and is found within the atom's nucleus. And then a third subatomic particle, this would be the particle that has no charge, is the neutron. And that is the particle with about the same mass as a proton, or the same weight as a proton, and it's uncharged electrically, and it thus is electrically neutral because it doesn't have neither a positive nor a negative charge. And that is also found within the nucleus along with the protons. Then we have two other terms here. We have those things that are ions, that is a particle that is electrically charged because it has either gained or lost one or more electrons, and we'll study specific types of ions. And then we have a molecule, which is a particle formed by the chemical union of two or more atoms. So we know that an atom is the smallest particle of an element, and the element is the basic chemical substance composed of atoms. Elements are represented by one or two letter symbols that are shown in the periodic table of elements, and you could see that in the appendix of the textbook. There are 116 elements that exist in nature. However, only approximately 26 are naturally occurring in humans. You can learn the elements and their chemical symbols by looking at the periodic table. The most abundant elements that are naturally occurring are those called carbon, abbreviated C, hydrogen, abbreviated capital H, oxygen with a chemical symbol O, and nitrogen with a capital letter N as its symbol. And these are the elements that are also frequently occurring in humans. So they would be CHON. But we also know that important to life are phosphorus and sulfur as well. So all matter is composed of elements. We have elements that are bulk elements, we have elements that are trace elements, and we have elements that are ultratrace. Compounds are composed of atoms of different types that are chemically bonded. So if we look at atomic structure, there are three subatomic particles, the proton, neutron, electron, and we already have gone over those. If you look here, this is the Bohr representation of the lithium atom. So you have the neutrons and protons in the middle, and the electrons will circle about the nucleus in what we call electron shells or orbitals. So the nucleus itself is the central part of the atom, and in that central part of the atom, it is composed of the protons and neutrons. Both of those have weight, and electrons move around the nucleus, and they have minimal weight. From the periodic table, you can look at the atomic number and the atomic weight. The atomic number is abbreviated A and then the hashtag symbol, so it would be atomic number, and that would be the number of protons in the nucleus of an atom. Each element has a unique atomic number, and if the element is not chemically combining with some other element or itself, you could say it equals the number of electrons as well in that particular atom. The atomic weight is the approximate number approximated by the number of protons plus the number of neutrons in one atom. Since a proton and a neutron each have approximate weight of 1, that is how we'll calculate the atomic weight. So typically when you look at an elemental block on the periodic table, the atomic number is just represented by that one number. It's one whole number. And the atomic weight is often represented by a number with some sort of decimal placements. So it could be, for example, 12.000. That would be the atomic number or the atomic weight for a particular atom. So if you look for hydrogen, the atomic number 1. For helium, the atomic number is 2. And for oxygen, the atomic number is weight. In continuing with the structure of matter, we have things called isotopes. So isotopes are atoms of an element that have the same atomic numbers but different atomic weights. That is because the same number of protons are there, but they have a different number of neutrons. So oxygen often forms isotopes, and you have oxygen 16, 17, and 18. Please note that the atomic number is the same for the atom that is, so oxygen 16 is the normal oxygen atom, but then that atomic number would be the same for the number of protons, but what changes there would be the atomic weight, because you're altering the atomic weight of that atom. So 16, 17, and 18 refers to atomic weights, and you will see those differently among the oxygen atoms. So basically, isotopes themselves, the nuclei of some isotopes are stable, but then you have the nuclei of other isotopes that are unstable and break apart to become more stable. When the nucleus of an atom begins to break apart, it releases radioactive energy, and this is where a radioactive isotope comes into play. Radioactive isotopes are unstable, releasing pieces of themselves in the form of atomic radiation. Radioactive isotopes have many biological uses. You can see radioactive isotopes used in anatomy and physiology. They can be used as tracers for diseases or problems, x-rays, so on and so forth. So for an element, the atomic weight is often considered the average of the atomic weight of its isotopes. Molecules and compounds. Molecules are particles formed when two or more atoms chemically combine. A molecular formula represents the numbers and kinds of atoms in a molecule. So if atoms of the same element combine, they will produce molecules of that atom. For example, oxygen gas, which is abbreviated O2, that is its molecular formula. So you have two oxygen atoms to create one oxygen gas molecule. The other one would be nitrogen gas, N2. If atoms of different elements combine, they form molecules of substances called compounds. So compounds would include water, H2O, and glucose, C6H12O6. But we also refer to these as molecules as well. You've heard of a molecule of glucose, you've heard of a molecule of water. So you can see molecular formulas below. These are depicted by the elements that represent the number of each atom present in a molecule. So in H2, that subscript 2 represents 2 hydrogen atoms. In the molecule of glucose, molecular formula C6H12O6, the subscripts there would represent 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms. And it's not located in the front there, but out in the front of this molecule, located right about here, you could put a coefficient, and that coefficient would tell you the number of glucose molecules you have. So for example, if we look at the molecular formula here for water, if I had 3 out front here, 3H2O, that coefficient would tell me that I have 3 water molecules, a total of 6 hydrogen atoms, and a total of 3 oxygen atoms. Even though there is no subscript there, that subscript is assumed to be 1, so you don't need to put it in. If it's greater than 1, as you see here, 6, 12, and 6, you would have to write that in there to indicate. So if we look at molecules and compounds, here we see a bunch of molecules. So here we have hydrogen gas and oxygen gas. Under electrolysis or some sort of electrical charge, you can form compounds of water, which would bring H2O together. And there you can see the structural formulas of these molecules and compounds. So back here we have molecular formulas. These would represent structural formulas, as you see them structurally drawn. So bonding in atoms. The electrons of an atom are arranged in orbits or shells or energy levels around each central nucleus, and the characteristic numbers of electrons fill each shell. So 2 electrons can fill the first shell, which is closest to the nucleus, and then 8 electrons can fill the second and third shells each, specifically. So if we look, electrons of an atom occupy the regions of space called electron shells. And for atoms with atomic numbers of 18 or less, the following rules apply. That first shell, 2 electrons, and each additional 8 electrons. So if we look here, we have hydrogen, helium, and lithium, the first 3 elements of the periodic table based upon their atomic number. Hydrogen has 1 proton, it has 1 electron with an atomic number of 1. Helium, atomic number 2, has 2 protons and 2 electrons. And lithium, atomic number 3, has 3 protons and 3 electrons. So you can see here, we have its 1 electron in its outermost shell. Here we have 2 with 2 electrons in that outermost shell. And then here we have 2 electrons in the first shell, but it has a total of 3 electrons altogether here in the lithium atom. So we have to put that last electron in the second shell. Sodium, do the example, sodium, abbreviated Na, has an atomic number of 11. So it has 11 protons and 11 electrons. Draw the sodium atom in your notebook. Now, why on earth would you want to know about electrons? Well, that comes down to bonding. And bonding is shown with, it's the force of attraction between atoms. And that force of attraction could occur in one of 2 ways. It depends on what's going on with the electrons, specifically the valence electrons. And valence electrons are electrons in the outermost shell. So when electrons are being transferred between atoms, you have ionic bonds forming. An ionic bond is an attraction between a cation and an anion, and it's formed when the electrons are transferred from a metal atom to a non-metal atom, and we'll go over all this in class as review. So here you see that sodium atom with its 11 protons and 11 electrons. So you see we have 2 electrons here, and then on the outside we have 1, 2, 3, 4, 5, 6, 7, 8. Over here we have chlorine. There's the chlorine anion because it has a negative charge. Here we have its full outer shell of electrons. So what took place here? What occurred? If you drew your sodium Bohr model, you would have saw that it would have had a first full inner shell. It had a full 8 electrons in its second shell, and then it would have a third orbital here with just one electron in it. If you draw the chlorine atom, it has 17 protons and 17 electrons. In its first shell, it had 2. In its second shell, it had 8. In the third shell, all right, it was missing 1 electron to be filled. So what happened was, instead of sodium gaining 7 electrons, chlorine gained sodium's 1 outermost electron, its 1 valence electrons. And that's where we got a positive and a negative charge here. So this would become an anion, or no wait, this would become the cation gaining a positive charge, having lost that 1 electron to chlorine, which will now become the anion. Together these are collectively called ions because they now have net electrical charges as atoms. So an ion is an atom that gains or loses electrons to become stable, an electrically charged atom. A cation or a cation is a positively charged ion, and it is formed when an atom loses an electron. In ionic bonds, that would be the metal atom. An anion is a negatively charged ion formed when an atom gains electrons. That would be the nonmetal. In covalent bonding and ionic bonding, ultimately what you want to happen, what you would like to see, is that atoms form bonds with other atoms to fill their outermost or valence shell energy levels. And that would be, in chemistry, you may have learned the octet rule. And basically what the octet rule states, that except for the first energy level, which contains 2 electrons, atoms react with other atoms, so they'll have 8 electrons in their valence shell. And that valence shell would be the outermost electron shell. So ionic bonds are formed when ions are created, whether it be a cation and an anion collectively, and that oppositely charged is the force of attraction between them. In covalent bonds, a covalent bond is formed by the equal sharing of electrons between atoms. Covalent bonds are very strong bonds, and you could have molecules forming, such as hydrogen and oxygen gas. Hydrogen, you can see here, we have 2 hydrogen atoms coming together, and they collectively will share their electrons. But we could also see it with oxygen and nitrogen. These molecules would form, and you would have 2 electrons being shared between the 2 electron pairs being shared between oxygen and oxygen. You'd have 3 electron pairs being shared between 2 nitrogen atoms. So this gives us the difference between a single covalent bond, a double covalent bond in oxygen gas, and a triple covalent bond here in nitrogen gas. Bonding of atoms, their structural formulas. So here you can see the single, double, and then here you see the single and double covalent bond for hydrogen gas and oxygen gas. Here's the molecular formula. Here's the structural formula. Here you can see the molecular formula for water. Here you can see its structural formula. And then here you see it for carbon dioxide gas. And here is its structural formula and molecular formula. So when you create a covalent molecule, such as hydrogen gas or oxygen gas, the electrons are typically shared evenly between those atoms. So those are what we call nonpolar covalent bonds. Sometimes what happens is, in a covalent compound, you can form a polar molecule. And in polar molecules, the molecules have a slightly negative end and a slightly positive end. And this will result when electrons are not shared equally in covalent bond formation. Water is an important polar molecule to life on Earth. So what happens here is oxygen has that greater electron pool. So electrons tend to spend more time around that oxygen atom than they do around the hydrogen atoms. And that creates slightly positive hydrogens and a slightly negative oxygen end. Now remember that these aren't full electrical charges, they are slight charges. When you get those slight charges on a water molecule, you can form a third type of bond called a hydrogen bond. And a hydrogen bond is a weak attraction that is formed between the positive end of a polar molecule and the negative end of another polar molecule. So here we see it formed between water molecules, and it's important for protein and nucleic acid structure as well. So interactions between water molecules and DNA chains, these bonds are easily broken. So these hydrogen bonds, because they are fairly weak, they can be easily broken and then easily restored as well. Picture a glass of water if you spill it, and that water is flowing on the floor or on a table surface or whatever. As it flows, those hydrogen bonds are constantly being broken and reformed between the water molecules that would make up that glass of water. So in understanding chemical bonding, both ionic and covalent, and then we add our few types of covalent bonds. We have the nonpolar, polar, and then from that we had hydrogen bond, which would be our third type of bond. We understand chemical reactions. And a chemical reaction itself occurs when chemical bonds form and break among atoms, ions, or molecules. And what happens is you could form a chemical equation. And reactants are the starting material of the reaction, the atom, ions, or molecules, and they can form the products, which are substances formed at the end of the chemical reaction. So there are three types of chemical reactions. The first type of chemical reaction is a synthesis reaction. In a synthesis reaction, you have the building of a large molecule, polymer, from smaller building blocks called monomers. So you have a constructive or anabolic reaction. Think of anabolic steroids. They build tissue. So in an anabolic reaction or a constructive reaction, you're going to create something. You're going to build it. Bonds are formed which now hold chemical energy. Water is usually removed from building blocks to form the bond. An example of this would be that dehydration reaction, dehydration synthesis, where you dehydrate the molecule to form water and then build the larger molecule called the polymer. Examples would be building of a large protein polymer from many smaller amino acid monomer subunits. The second type of reaction is breaking a large molecule, a polymer, down into its building blocks, which would be its monomer subunits. That is a decomposition reaction. So in this, you're going from polymer to monomer. In this, it's a catabolic reaction or destructive reaction. Usually reactions in the digestive system are decomposition reaction because you're breaking things down. And when you break things down, you're going to be breaking bonds, and therefore you're going to release chemical energy, which would be an exergonic reaction. So you have energy going out instead of energy going in, which is endergonic. In this case, water is going to be used to break those bonds. That would be hydrolysis reaction. So when you go from that polymer to those monomer subunits, and we'll look at this in biochemistry, or if you can recall it from biology, water is broken, and it goes in and it breaks the bonds between the polymer molecule that is holding all those monomers together, and then you'll individually form each of those monomer subunits. The third type of reaction is an exchange reaction, and it involves degradation followed by synthesis. So the products can change back to reactions. So you could have many chemical reactions are reversible reactions, and the products can be changed back and forth from reactant to product. In acid-base chemistry, you have acids, bases, and salts. These ions are referred to as electrolytes or charged particles. So electrolytes are substances that are going to release ions in water, such as salt. So you form the sodium cation and the chlorine anion. Electrolytes must be maintained within a very narrow range in our blood and tissues. That would be homeostasis, playing a role there, that stable state for electrolyte balance. Electrolytes are needed for muscle contraction, they are needed for nerve impulses, they are needed for bone growth, etc., etc. Examples of electrolytes that we would see are sodium, Na+, potassium, K+, chlorine, Cl-, calcium, Ca+, the phosphate, PO4 negative, and then you have some others in there that we'll learn along the way. Acids are molecules or electrolytes that are going to dissociate to release hydrogen ions in water. So acid dissociation, you form that hydrogen cation, H+, and an anion. So an example would be chlorine and hydrogen. They form hydrochloric acid, HCl. When you put HCl into water, you form H+, you donate that proton, you also form Cl-. On the reverse end of the spectrum of the pH scale, you have bases, and bases are substances that are going to release ions that combine with hydrogen ions, and that's going to make it more alkaline. So often that ion would be the hydroxyl anion, which is OH-. And in reverse of that, you would also form a cation. So when you add sodium hydroxide to water, NaOH, which is a strong base, what will form is NaOH, and then you'll form Na+, that will be your cation, but you have this hydroxyl anion in there that is going to bind with NH+, and you'll form water. So you will tend to neutralize the acid and base there. And when you neutralize an acid and a base, what happens is you form water and salt. So salts are electrolytes formed by the reaction between an acid and a base. So salts dissociate and ionize into ions when dissolved in water, and an anion is formed and a cation is formed. So basically here you could see if you were to add 3 drops of HCl to 3 drops of NaOH, you'd form water and NaCl, where the OH of the sodium hydroxide will bind with the H of hydrochloric acid, forming water, and then you have the sodium from sodium hydroxide bonding with the chlorine atom of the hydrochloric acid to form the salt, NaCl. And that is basically acid-base chemistry. And then you could see here that salt being disassociated in a molecule of water, and how those molecules of water would form around the cation, the sodium cation, and how the molecules of water would also form around the chlorine anion of that salt crystal. And lastly, this takes us to, when we talk about acids and bases, our pH scale, so acid and base concentration. And we know that the pH scale, it's the relative concentration of hydrogen atoms and hydroxyl ions determine the pH in our blood, fluids, and tissues. The pH in the body is equal, so the hydrogen is equal to the amount of hydrogen ions plus the amount of hydroxyl ions in solution. Basically, what does pH measure? pH is a measurement that is equal to the negative log of hydrogen ions in solution. So the equation for pH is equal to the negative log of the hydrogen ions concentrated in solution. And then you have that pH scale that ranges from 0 to 14. where if the hydrogen ions are greater than the hydroxide ions, you're going to have an acidic solution. On the other end of the spectrum, if the hydroxyl ions are greater than the hydrogen ions, you will have a basic solution. And then a neutral solution is when the hydrogen ions are equal to the hydroxyl ions, and that would be a neutral solution. And you could see the definitions here for all of that. And then you could see common substances and their pHs on the pH scale. So blue would represent that of an alkaline or basic solution, and red would represent that of an acidic solution or an acid. And as you could see here, right around here is where they are neutral. As you increase the pH, you are becoming more alkaline. As you decrease the pH, you are becoming more acidic. So this would mean that the hydrogen ion is greater in solution. Over here, the hydroxyl ion is greater in solution. As far as blood, blood pH, its normal pH is goes between 7.35 and 7.45. So physiological, we're looking at 7.4 for a pH. If the pH is less than 7.4, you have acidosis occurring, and acidosis is lethal below 7.0. So acidosis is when the blood pH drops to 7.0 and 7.3. Below 7.0, it is lethal. Alkalosis is on the other end of the spectrum, and that's when the pH is greater than 7.4. So alkalosis is 7.5 to 7.8. And it is lethal above that 7.8 range. So how do we maintain a normal blood pH between 7.35 and 7.45? That would be where homeostasis comes in, and you have homeostatic mechanisms that are going to help regulate the pH, which would be using buffers. So you have this buffering system, and we'll look at this a little bit more in chapter 19. So and also in chapter 21, we'll study buffer systems in blood and cardiovascular system as related to how they work. But ultimately, for now, our definition of a buffer, buffers are composed of both weak acids and weak bases. So buffers are compounds that are added to solutions that are going to prevent sudden changes in pH. And sudden changes in pH, it's usually weak acids. It functions by donating a hydrogen ion when needed and accepting a hydrogen ion when in excess. They are very important to biological systems because they prevent those abrupt changes in pH. An example would be carbonic acid, H2CO3, which would serve as a buffering system. So when the pH is rising equation, it goes to the right. When pH is falling, the equation will go to the left. So for example, what you'd have there in the equation as a reactant that can go in reverse from reactant to product, you have H2CO3, which is carbonic acid, will form the HTCO3 anion plus that hydrogen cation, and that reaction can go reverse between the carbonic acid H plus donor to the bicarbonate ion and the hydrogen ion. So that's it for sections 2.1 and 2.2. Our last section of this chapter will be the next lecture, which will be section 2.3.